So thanks so much for this opportunity. It's really nice to give other talk. I actually gave a talk uh, in the first conference, and it's quite nice to see how it's increasing over the time. We have more than 40 participants, and so it's quite nice to see how it's increasing. So my talk is about the effect of inducers, hydrophobic inducers on biosurfactant production. But more than that, I would like to talk about agro-industrial wastes. Have you considered how much agro-industrial waste you have on tomato process, carrot process, and so on? So it's a huge amount of nutrients and a plenty of possibilities to work with. In this topic, I'm gonna try to show one possibility of cassava waste, which is cassava wastewater, in order to produce biosurfactants. When you heard about biosurfactants, just think about it, soap, a detergent structure, but that was produced by living cells, in particular bacteria. So who do you say am I? I'm food engineering. After that, I graduated in Master Food Science, and then straight for I started the PhD program, in which I did the split PhD in England, University of Reading. After that, I worked as a research in the chemical engineering department and so on. I have started to work as a professor in the chemical and food engineering department of Federal University of Santa Catarina. I do also work as a professor in the postgraduate program in chemical engineering, which is one of the finest program in Brazil in chemical engineering. So the summary is cassava and concept of green chemistry. Just a brief introduction, biosurfactant production, effect of hydrophobic inducer on the production of biosurfactants, current works, and conclusion and perspectives. So when you think about cassava production, when you sum up Nigeria, Republic of the Congo, Thailand, Ghana, and Brazil, they together represent more than half of the global production. And I keep wondering myself why India and China, they don't make part of that, because they have a really huge population. Actually, this kind of crop is related with low-income population. And one of the main products is the cassava flour. So the whole process of a cassava flour is quite simple. The first you have to crop that, and after that you have to wash. And this wash water is one of the residue, the waste that we can apply in order to aggregate its value. So after wash, you have to cut and peel. And the cassava peel by itself is one of the waste. In fact, we have produced a kind of prebiotic compound, which is named oligosaccharide, using cassava peel. To the best of my knowledge, is the first article on this topic, and it is under review right now. So after cassava peel, you have to grind that and dry. When you re-dry that, I mean, you have to press that in order to remove liquid from that biomass. And this is the most special residue for us. It's named cassava wastewater. It's quite rich in nutrients, in sugar, carbon, and nitrogen, phosphorus, magnesium, and a plenty of nutrients. And from that biomass, you have to do some thermal treatment, and after that, you got your cassava flour, which is quite tasty, personally speaking. So when you think about the whole process, 1 kilogram of cassava root leads to more than half kilo of solid waste, and I mean cassava peel, and more than 6 liters of wastewater when you sum up this wastewater and cassava wastewater. So it's too much. In fact, currently, it is a problem to the industry because they have to treat that and after dispose of that. So we have to propose a project in order to use these residues, correlating them with a biotechnological process. And we submitted that in 2019 to a FAPESP, which was a call, and we won more than $100,000 to run this project. So for start, what is the concept of green chemistry? In general terms, green chemistry is the design of chemical products and processes that reduce or eliminate the use of or generation of hazard substance. Green chemistry apply across the life cycle of a chemical product, including its design, manufacture, using, and ultimate disposal. Green chemistry is also known as a sustainable chemistry. So, in general terms, if you try to reduce the toxicity of a traditional process, or just partially to reduce the impact of the environment of a process or a product, you are doing green chemistry. You are applying green chemistry. So we started this project try to relate green chemistry and cassava. As you can see, you use it scope database in order to identify scientific trends. It's quite nice. I really like to mention this kind of opportunity, this kind of possibility in order to you identify the scientific trends of your subject. So as you can see here, green chemistry is sharply increasing, and also cassava is sharply increasing. However, when we split, we divided cassava in specific areas, the most of agriculture, the most papers or scientific data related with cassava is correlated with agriculture. I mean, increasing the cropping, the production, and so on. Just a small part is related with chemistry, engineering, or even biochemistry. This project maybe is correlated with chemistry, engineering, and also biochemistry, personally speaking. So our project are trying to move a traditional industry to a quite new and sustainable industry. I mean, change from a refinery to a biorefinery. And these both products, xyloliposaccharides from cassava peel and maybe biosurfactants from cassava wastewater is economically attractive. And also, and again, there's a plenty of possibility to work on. So the whole process is the first subject is try to describe the cassava wastewater. Cassava wastewater looks like this, liquid. As you can see, it's quite dense, it's quite yellow. There was a small odor flavors, I mean. And again, it is now a problem for the industry because they have to treat that in order to dispose of that. So for them, it's like a problem. For us, it's like a solution because it's a low cost or medium, and actually is free cost culture medium. And when you try to relate economically with biotechnological process, the culture medium represents more or less 30% of the total cost production. So it's too much, and for us, this is for free. But before to start the fermentation process by itself, we have to take these cassavas to water, and we have to boil that, and after that you have to centrifuge that. We have to centrifuge that in order to remove starch from that solution. And after that, of course, the starch is another possibility to for instance, to produce edible fumes, or anyway, any other possibility. So after that, we start the biosurfactant production. So it's a quite different biotechnological process because there's a lot of foam producing. We can recover this foam by the top of the bioreactor, and after that, we can run some purification process, and this purification process, the most prominent purification process is the ultrafiltration, which is membrane-based step. And with that purified product, we have plenty of opportunities to apply in pharmaceuticals, for instance, in food, and but personally speaking, I think one of the most promising application is in the agriculture. I mean, today we have been consuming plenty of pesticides, and we can apply that that kind of compound in the agriculture and change, and even partially we can change the pesticide for eco-friendly pesticides as a surfactant. Surfactant is one of the biosurfactants, and they are composed, it is composed, sorry, for 7 amino acids and 1 fatty acids. And personally speaking, it is a remarkable structure. If you can see here, there's a deconfiguration. The deconfiguration is quite hard to find in nature. You can find in some insects, but regarding to bacteria and so on, is really hard to find the deconfiguration. And there are 7 amino acids here, but they can change. I mean, if you change the bacteria, if you change the culture medium, if you change the biotechnological process parameters, agitation, rotation, temperature, and so on, you can change this kind of amino acids. And this is that the research is about. So looks like a really master plan when you think about is a free culture medium. We can use that for biotechnological process, biosurfactant production, and so on. But in 2016, we have identified that for one hand, okay, it's a free culture medium. On the other hand, there are too much protein in this cassava to water. And the protein is may hamper the purification process when you think about ultrafiltration. So from this point, we have been trying to overcome this problem. And there's a lot of publication on this in this subject. The most recent from our research group is try to use ceramic membranes to purify the surfactant, and also to use some inducers in order to enhance the production of surfactant. This is a quite nice publication also. Is a glycolipid, but is the process is basically the same. You use it cassava water as a low-cost culture medium, and this one is the simultaneous production of surfactant and one flavor, 2,3-butanediol. And regarding the application of biosurfactants in agriculture, this chapter book groups a really nice information on it, because you have activity against phytopathogenic fungi, but we have also observed that some biosurfactants have some kind of biostimulant effect on agriculture seeds. And regarding cassava water by itself, this chapter book just compilate the possibility of use cassava water to produce pigment, to produce protein, to produce flavors, using yeast, bacteria, microalgae. So virtually any microorganisms can use cassava water as a culture medium. So it's quite interesting. So we have been trying using some specific inducers like oleic acid, palmitic acid, soybean oil, and also the control a different concentration. And in order to see how they improve, if they improve the productivity of biosurfactant, but mainly if they induce some kind of change in this structure. When I mean change, I mean virtually we can change these amino acids for other amino acids, and we can observe a really new biotechnological property, anti-cancer or anyway. So in order to identify this change, all addition of the inducer, oleic acid, palmitic acid, and so on, have changed the microbial growth, as you can see here. And it changed also in the pH, sorry, and surface tension, which is a direct measurement of biosurfactant produced. So there's a really strong effect when you use some inducers with cassava water as a low-cost medium. And after that, we have used Malthoff MS, which is a spectrometry instrument facility to identify this kind of structure. I mean, we use Malthoff MS in order to evaluate it if the inducer had changed some amino acids. And our research group has write up a program. We have we hope to publish that maybe next month, in order to collect this data and try to identify if this data is correlated with new structure of surfactant. So I'm gonna give you a sample of our results. We don't have all results so far analyzed, but for instance, this is the control. I mean, it's the fermentation process with no inducer at all. And this one is the palmitic acid. That means it's the biotechnological process with cassava water as low-cost medium and with palmitic acid. So we can see here there's a 16, 10, 16, 10, 16. So it's basically the same. 10, 30, 10, 30 is basically the same. 10, 40, 40, 10, 40, 40 is basically the same. This Malthoff MS data, right? However, we look it up here is 10, 16. And 10, 16 could be this chemical structure. We didn't see this 10, 16 in the control. So it maybe is a kind of new chemical structure of biosurfactants. Now we have to further to purify that in HPLC and run a double MS. I mean, to analyze that by MS and also another MS. Finally, I would like to thank you, my research team. We have a postdoctoral research, we have a master student, doctor student, doctor student, doctor student, research, postdoctoral research, and my professor colleague also. They do all the work, and I'm really thankful for them for their hard work. As a prospection, as a conclusion, we have best hopes in trying to identify new application of biosurfactants and new chemical structure. We think we are doing well with purification process, application process, and really fine chemical identification of this structure. And we may change even partially the agriculture, because of course pesticides is related with a plenty of diseases like cancer and so on. So one of time we have to change that. I don't know if it's in 5 years, in 10 years, but we have to change that. And finally, would you like to apply the concept of green chemistry to food industry? There's plenty of other possibilities, as I already mentioned, carrots, tomatoes, and potatoes. Any food can be related with green chemistry, and I thank you all for your attention, and I finalize my talk here.